This tutorial shows the handling of HibarRT HPLC columns, the mounting of a pre-column or guard column holder, and the easy connection to any HPLC instrument. The column box contains not only the column itself, but also the certificate of analysis and, in addition, the very useful insert sheet. This insert sheet contains important general information about the column and recommendations for column care and use, including, for example, column equilibration instructions. Each single column is individually tested and is packed with its own unique certificate of analysis. The individual column number is printed both on the certificate of analysis as well as on the column label. HibarRT columns are available with many different column lengths and internal diameters. Pre-columns or guard columns are often used to protect HPLC columns from contamination from the sample matrix and to extend the lifetime of the column. To minimize extra column effects, the connection of the pre-column to the analytical HPLC column should be dead volume free. For direct connection of LicroCart 4x4mm guard column cartridges to HibarRT columns, a reusable pre-column holder is available. The same pre-column holder fits HibarRT columns with internal diameters of 2, 3, 3.9, 4, or 4.6 millimeters and lengths of 75, 100, 125, 150, 250, or 300 millimeters. To mount the pre-column holder to a HibarRT column, first remove the column end fitting at the column head using a wrench. Next, screw the end fitting nut of the pre-column holder onto the column head and tighten with the wrench. Be careful not to over-tighten the nut. Place the LicroCart 4x4mm guard cartridge into the holder with the narrow end first. Place the end fitting cap over the nut and tighten firmly using the wrench. Now the column is ready for connection to any HPLC instrument. All standard connection systems can be used. The HibarRT columns should be installed in the flow direction of the system. The capillary coming from the pump and sample injector is connected to the column head, and the capillary tube from the detector is connected to the column end. Flexible fittings are self-adjusting and allow all kinds of columns to be used with the same HPLC instrument without changing the connection tubing. Also, flexible hand-tight knurled nuts with flexible capillaries, such as peak capillaries, fit without any limitation. Traditional stainless steel capillaries with ferrules sometimes need to be renewed when changing between columns from different manufacturers to eliminate possible dead volume. The connection of a column together with guard column to the HPLC instrument follows exactly the same rules as described already for columns without guard columns. There is no difference. Once the HPLC column is connected to the instrument, it is ready for equilibration. HPLC columns are commonly equilibrated first with 100% organic solvent, such as acetonitrile. For best results, it is strongly recommended to use a high-quality solvent such as Lycrosolve Gradient Grade. Changing a pre-column in front of the HibarRT column is very simple. Just stop the HPLC pump, open the finger-tight pre-column holder, and replace the old pre-column with a new one before closing the holder finger-tight or using a wrench. If the pre-columns are stored in a small bottle with organic solvent, like acetonitrile, the equilibration step for the pre-column is not needed. To be your partner in chromatography, that's our goal at Merck HibarRT. As one of the leading manufacturers in liquid chromatography, we are committed to future developments of sorbents, analytical and preparative columns, sample preparation, thin layer chromatography, and biochromatography products.